Good morning. I'd first like to thank the organisers for the opportunity to speak at this fantastic meeting. I'm Joe Grove. I'm based at the Institute of Immunity and Transplantation at UCL, and I'm interested in virus entry. And I'm investigating this using a range of techniques, including basic virology, but also advanced quantitative imaging. And it's this that I'm going to talk to you about today. Now, as a virologist, I'm presented with a fundamental problem, and that is that viruses are really small. And I know this sounds like I'm stating the obvious, but I think it's worth reconsidering for a moment. So a typical virus particle is maybe 50, 100, 200 nanometers across. A nanometer is a billionth of a meter. The human mind, or at least my mind, isn't very well equipped to deal with these kind of numbers, so I resort to analogy. Imagine we could make the world a lot bigger, so much bigger that a virus particle is now the same size as an apple. In this giant world with apple-sized viruses, I would be around the same length as France, and my thumbnail will be around the same size as Paris. So I think this gives you some illustration of the huge size differential between the world that we inhabit and the world inhabited by viruses. Viruses exist at a molecular scale, and this presents a practical problem when applying microscopy. If we consider a 100 nanometer virus particle that's decorated with fluorophores, when viewed down a microscope, it appears as a spot to our eye or to the detector of around 200 to 500 nanometers across. So that spot doesn't well represent the true structure. This isn't necessarily a problem if the virus particles are well spatially separated. So for instance, here, in this artificial example, I can count that there are 8 viruses and know something about their relative position to one another. However, as they get closer together, their signals begin to merge and that information is lost. Now I can illustrate this further by an example of a nanoscale word that's decorated with fluorophores. If we view this by conventional microscopy, due to this so-called diffraction limit, we have no chance of reading the word. However, super-resolution microscopy allows us to break through this diffraction limit. And I am largely using a technique called localization microscopy, which was principally developed by our keynote speaker, Eric Betzig. And I just want to talk briefly about how this works. So localization microscopy uses various photochemical tricks to ensure that spatially crowded fluorophores do not emit light simultaneously. This allows them to be analyzed in isolation. So let me illustrate this by going back to this example of a nanoscale word. If we have an individual fluorophore on this emitting light, it will appear as a diffraction-limited spot. Using mathematical fitting, we can estimate the true position of this fluorophore with a high precision. And through multiple rounds of localization and mapping, we can reconstruct this nanoscale word and read it. So now to go to a real-world example, this is a cell expressing CCR5, a co-receptor for HIV. I've labeled it with an antibody directly conjugated to a fluorophore and viewed it by conventional microscopy. And I think you can see that there's only really a homogeneous sea of green apparent. It's very difficult to pick out any detail. However, if we go to localization microscopy mode, we can induce the fluorophores to blink on and off. And then using these spatially isolated diffraction-limited spots, we can map the individual positions of each of the molecules and reconstruct the nanoscale organization of this receptor. As it turns out, this data can be applied to all sorts of images. This is a video taken by my friend and colleague Ricardo Enriquez, who some of you may know as he was previously based at the Institut Pasteur. He applied his quick palm algorithm to this to show that he could indeed reconstruct the Eiffel Tower. Okay, thanks to super resolution microscopy, it's a very exciting time to be a virologist, because for the first time, we can take a true virus eye view of the cell. These images are taken from studies that use localization microscopy to study HIV. This is from the Lippincott-Schwartz lab. It shows TSG101, a host component in green, inside a budding HIV particle. Importantly, this gives us insight into the way that the host escort machinery contributes to membrane scission during HIV release. This image is taken from a study from the Zimmer lab based at the Institut Pasteur. And they studied the change in size of the HIV capsids as they undergo nuclear import. I want to demonstrate how we're using these techniques, but to do this and to give you a complete story, I'm going to step away from pure virology and talk about the cell biology of the plasma membrane that underpins virus entry. So prior to the development of super-resolution microscopy, the best way to get a virus eye view of the cell was using electron microscopy, as illustrated by this scanning electron micrograph of some beautiful HeLa cells in culture. I'm interested in virus entry. The action here is occurring at the cell surface, and there are a number of EM techniques that allow us to take a virus eye view of the plasma membrane. For instance, we can take cells grown in culture and hit them with a blast from a sonicating probe or a jet of buffer and remove their cell bodies, just leaving the adherent plasma membrane intact. This can then be viewed by electron microscopy en face, and that's what's shown here. This is an adherent plasma membrane sheet from a HeLa cell, and we're looking now onto the cytoplasmic face of that membrane. And I'm going to play a video in which we zoom into this surface, and I think you'll agree that there's some eye-catching features apparent. Okay, so aside from these relatively featureless areas of plasma membrane, we can also see these areas covered in this flat hexagonal lattice, and also these electron-dense spherical structures. As some of you may know, these represent different manifestations of the Clathrin endocytic apparatus. These are classical Clathrin-coated pits or vesicles, whereas these are flat Clathrin lattices. Clathrin-mediated endocytosis is the principal mechanism of endocytosis for eukaryotic cells, and it is essential for many functions occurring at the plasma membrane, such as signaling, and is also necessary for the entry of a number of viruses. I'm going to illustrate how this process works by playing a short video that uses molecular data to reconstruct the events of Clathrin-mediated endocytosis. And this is pretty much in real time. So in this animation, we're going to be coming through the plasma membrane to look at the cytoplasmic face, such as we were just viewing by EM. So these are receptors in the plasma membrane, and now we're looking at the cytoplasmic face. And in green, we can see these adapter complexes, the AP2 complex. These interact with the cytoplasmic tails of receptors, and also recruit Clathrin trischelia, which assemble into cages around an invaginating Clathrin-coated pit. This can recruit endocytic cargo, and then as the cage undergoes increasing assembly, you lead to a spherical structure that's connected by a thin membranous neck. This then undergoes scission by the small GTPase dynamin, which severs the vesicle. The Clathrin cage can then disassemble, and the vesicle delivers the cargo to the endosomal network. What's strikingly absent from this video is any of these flat Clathrin lattices. And that's because this is the accepted standard model of Clathrin-mediated endocytosis, but very little is known about how these larger structures contribute. So how pits and vesicles contribute to nutrient uptake, signal regulation, and virus entry is pretty well known. And this is reflected in the number of PubMed citations for clathrin-coated pits or vesicles. The role of these flat lattices is really unclear. And actually, this is also reflected in the paltry number of citations for these structures. So we set out to use super-resolution microscopy and other quantitative techniques to describe these flat clathrin lattices and better understand their contribution to plasma membrane biology. So first, we started by using electron microscopy to take a survey of clathrin heterogeneity in various cell types. We prepared membrane sheets from the bottom and the tops of cells, and then took multiple images, and I manually segmented these, which is a little time-consuming, by just drawing around each of these continuous clathrin structures. This then allowed us to extract the size and shape characteristics of these structures and plot them. And that's what's shown here. In this scatter plot, each black dot represents an individual structure, and I'm showing the circularity versus their surface area, the two-dimensional surface area, in nanometers squared. You may notice there's this discrete population here of very round, relatively small structures. These represent the classical clathrin-coated pits and vesicles. But you also have this spectrum of increasingly large, increasingly regular structures that represent these pleomorphic lattices. Interestingly, if we look in other cell types, these lattices are pretty much missing, and we only see pits and vesicles. I'm going to cut to the chase. We don't know why this is yet. So there's likely to be a discrete molecular determinant of these different clathrin structures, but we don't know what it is. There's a caveat to this data, and that's that we've had to literally rip open the cells to get at these samples. So we wanted to view this in a more a non-invasive way, so naturally turned to fluorescence microscopy. However, conventional microscopy does not afford us the resolution to differentiate these different structures. Therefore, we turn to localization microscopy, and these are images of antibody-labeled clathrin at the adherent surface of HEC293T and HeLa cells. These insets here show the diffraction-limited image, and these are the super-resolution reconstructions. And what should be apparent is in the HEC293Ts, we have this punctate staining, which is consistent with only having these small round structures, whereas in HeLa cells, we see these ribbons of clathrin representing the lattices. And again, we can quantify this. Thankfully, I didn't have to manually do this. We can automatically do it, and do it to many thousands of structures across many cells to get a really in-depth survey of what's going on. And we pretty much find the same thing, and that is that in HEC293Ts, you almost exclusively have these small round structures, and the larger ones are relatively rare, whereas in the HeLa cells, we have these much larger structures. So in the HeLa, we can estimate that these large structures account for 80% of all the clathrin associated with membranes. And this accounts for around 10% of the plasma membrane. So this is a significant proportion. So having looked at their morphology, we then went on to study their dynamics using live fluorescence microscopy. We took HeLa cells expressing fluorescently labeled clathrin and fluorescently labeled dynamin. I remind you that dynamin is the GTPase necessary for membrane scission during vesicle budding. We thought that this would afford us a view of budding events occurring at clathrin pits, and maybe from these lattices as well, because we see budding profiles at the edge of these lattices. And what we saw was somewhat surprising. So in magenta, we have clathrin on the adherent surface of HeLa cells, and these large, bright structures represent the flat clathrin lattices. Surprisingly, dynamin is constitutively associated with these. So this is at odds with the canonical model of clathrin-mediated endocytosis. We viewed this by conventional, by turf microscopy. I took a frame every 3 seconds for 10 minutes. And I'm sure you can appreciate there's very little going on. These structures just sit there. And actually, I viewed these in independent experiments for an hour or two, and they just stay there for prolonged amounts of time. Now, some of the keen-eyed amongst you may notice in these empty areas of membrane, we can see diffraction-limited events that come and go. These are classical clathrin-coated pits, and we'll see one of these events in this zoomed-in video here. So we have a pit that appears in the center here, and then disappears with a coordinate spike in dynamin. So this is consistent with the model that was demonstrated in that animation I showed you. We could also analyze this data using chymographs. So here we're looking at the fluorescence trace at a piece of the membrane over time. And you can see for a clathrin pit, we have gradual increase in the clathrin signal, which indicates the assembly of this cage. And then this disappears with a coordinate burst in dynamin, which indicates the recruitment of dynamin to the neck to undergo membrane scission. Whereas these flat clathrin lattices have constitutive signal for both of these. We performed tracking analysis to quantify this data, and we could see around 10% of all the clathrin structures are these stable flat clathrin lattices. Okay, so these structures have a unique geometry. They are structurally distinct from the canonical structure. They're relatively abundant. In at least some cell types, they account for 80% of the membrane-associated clathrin and cover 10% of the membrane. And they're very structurally stable. So now we wanted to try and understand what consequence these have for the biology of the plasma membrane. And to do this, we turn to CCR5. As I said earlier, this is a co-receptor for HIV, but its physiological role is in signaling for chemotaxis in T cells and macrophage. So CCR5 is a chemokine receptor. It is a G protein coupled receptor. And upon addition of one of its ligands, CCR5, it initiates G-alpha I signaling and then recruits beta-arrestin, which sequesters CCR5 into clathrin structures for endocytosis. And this is how the cell surface level and signaling of this receptor is regulated. So we studied this in relation to our clathrin structures. So again, we have clathrin in magenta, and then antibody labeled CCR5 in green. And then this box here is just zoomed in these panels here. I took a frame every 30 seconds for an hour, directly prior to treatment with ligand. And you should be able to see over time that CCR5 redistributes to these large, stable, flat clathrin lattice structures, as shown here. And we can quantify this. And also, by looking at individual chymographs at a higher temporal resolution, we can see that CCR5 is being incorporated into classical transient clathrin-coated pits, but it is also constitutively associating with these flat clathrin lattices. So having studied this in live cells, we then returned to super-resolution microscopy to get a molecular view of these processes. So again, CCR5 is in green, clathrin in magenta. In untreated cells, CCR5 is relatively evenly distributed and doesn't show any or very limited association with clathrin. However, upon treatment, we see a distinct clustering of CCR5 in and around these clathrin structures. One of the many benefits of localization microscopy is the way that it provides data. So we have point coordinates that represent the molecular localizations of these receptors, and these are amenable to spatial statistics analysis. And I performed Hopkins analysis on this. Now, Hopkins analysis allows us to look at the co-distribution of CCR5 and flat clathrin lattices. And a Hopkins value of 0.5 would indicate a random co-distribution. A Hopkins value below 0.5 indicates that these two components are segregated, whereas above 0.5 indicates association. We have here some random simulations as control, and you can see that these give values of 0.5, which is consistent with a random distribution and shows that our algorithm is working. Unsurprisingly, in the presence of ligand, we see distinct association of these two structures, of the receptor and the lattices. Surprisingly, in the untreated cells, it would seem that these flat lattices can exclude receptor. So this would suggest that these lattices provide a novel means of segregating components of the plasma membrane. So we would propose that these lattices are able to compartmentalize events occurring at the cell surface. And this is supported by some other recent studies that came out around the same time as our work. So this work from a group based here in Paris demonstrate that flat clathrin lattices are necessary for sarcomere formation and for the anchoring of actin filaments in muscle fibers. And without clathrin, the muscle tissue is impaired. And then this work from a group in Yale demonstrate these large clathrin structures are necessary for the assembly of signaling complexes in Wnt signaling. So we would propose that flat clathrin lattices represent stable platforms for the organization of the plasma membrane. And we are interested in the future to understand what implications this has for membrane signaling, but also for virus entry. So it just leaves me to thank a number of people. So first, Mark Marsh. This work was principally conducted in his lab during my postdoc. I'm now setting up my independent lab at UCL, where I'm using some of these skills to study other aspects of virus entry and cell biology. Also, a number of people at the National Physical Laboratories, where I was initially trained in super resolution, and also the Lippincott-Schwartz lab at the NIH, where I did much of the two-color localization microscopy, and Ricardo Enriquez also at UCL. And I have to thank the various people who have funded this work and are funding me now to set up my lab. And thank you all for your time.